Guys, welcome back. Third part of protein, and now we are starting with the fourth part in protein, and we are beginning with the beta pleated structure of protein here. Okay, we have completed. We had started with the structure of protein in the last session where we discussed the alpha helix structure, isn't it? And now we are going to start with the beta pleated structure here, and then we'll discuss the structure of collagen and hemoglobin. If time permits, we'll discuss the structure of hemoglobin as well, but otherwise we'll discuss the structure of collagen. Okay, do let me know if I'm visible and audible to all of you. Let me just check if the live stream has started, and then we can begin. Yeah. Okay. So I'm your educator for biochemistry. Currently, we are conducting a session that is based on comprehension. We are going to take each topic slowly. We are going to dissect the topic into subtopics and cover the entire course of biochemistry. We have just finished with the super fast crash course of biochemistry as well as a rapid revision series in biochemistry, where we focused on the entire syllabus of biochemistry and especially on the important and the difficult topics, difficult to understand topics. Now we are going to take even smaller topics into account, and we are going to read up each and everything in detail, okay, as we are taught in our medical colleges. Okay, right? This is going to steer you ahead of your colleagues, and I'm going to focus, especially hi Jatin, I'm going to focus on people who are going to appear for their professional examination. So the next classes, the next week that I'm going to start, next week I'll keep classes specially dedicated for you people who are going to appear in professional examinations. Okay, you can give me Jatin wants classes on metabolism. Okay, so I'll keep a marathon. I'll try and keep a marathon on uh, metabolism. That marathon I'll be keeping in the Unacademy app. Okay, that marathon will be on the Unacademy app, Jatin, because here on YouTube there are time constraints. We cannot put long videos here. So that marathon session I'll put on Unacademy app. You can attend that. That will be absolutely free. A special class. Okay, that will be a special class, absolutely free. You just have to check my profile out and you will find. Okay, if you're following me, you'll get the notification. I'll publish that class probably in a few days. Okay, marathon series of metabolism for you to revise, because you would already be knowing that by now because you're going to appear in your professional examination. It is just for you to revise. Okay, next. Let us quickly discuss about the types of subscriptions available on Unacademy for NEET PG, the Iconic and the Plus subscription. Okay, you guys would be knowing about Plus, how you can access video lectures from top educators across the country, anywhere, wherever, even while traveling, even while sitting at your home, even while attending your college, wherever you want, you can attend that on your mobile, laptop, tablet, wherever. Okay, the Iconic subscription also provides you with access to resources from Prep Ladder. Therefore, it's called best from two of the best, isn't it? And while you can, after applying the educator code DRSHUKLA, Dr. Shukla, you can avail 10% discount. But if you apply this code DRSHUKLA on the 4th, that is today, and the 5th of January, you're going to get an additional 22% off. So can you see? You can fetch the subscription in peanuts, isn't it? Because this 36,000 for 2 years, 36,000 is for 2 years. And on this, you're going to get 22% discount. So 10% is how much? 3,600. 20% will be 7,200. And then 2% will be again 720 more. Okay, so it will be almost 10,000 will be reduced. So 2 year subscription in almost 27,000 you're going to get. Okay, isn't it the cheapest? Okay, cheapest available. No one can provide you this much 2 year subscription with so many things. You will get access to all the teachers in different subjects, in all the subjects of MBBS. Okay, and that too for just around 27,000 for 2 years. Imagine, in the plus subscription. Okay, so do give it a thought. If you're willing to subscribe, then I would advise you to do it within these 2 days itself. It will fetch you a lot of discount. Okay, now so we have completed the structure of protein. We have not completed. We have started with the structure of protein. We have completed the basic properties of amino acids. Okay, different types of amino acids. The synthesis of amino acids also we have done yesterday. And tonight we are going to complete the structure of proteins. Okay, we have discussed what is the difference between conformation and configuration. We have discussed what are the 4 orders of protein structure: primary, secondary, tertiary, quaternary, isn't it? We have discussed in detail the secondary structure. Do try and make this diagram. Try and make the atoms of nitrogen and carbon separately in different colors. Try and do mention what the pitch is, how many residues are there in per pitch, okay, in a normal alpha helix structure. And always remember, it is stabilized by hydrogen bonds mainly, and van der Waals interactions also increase the compactness of the core. Okay. Next is the beta pleated structure. Okay, why beta? Because it is the second structure to be discovered. The first one to be discovered was termed alpha. Now the second one to be discovered is termed beta. Why pleated? Because it appears as pleats. Pleats, you see the pleats of the sari. Okay, the pleats of the sari that women wear. Similarly, this is a pleat or a sheet-like structure. Okay, beta pleated structure. Okay, how does it appear? When you look at it end on, that is from one end. When you look at the structure, it appears zigzag or pleated, like the pleats of a sari. Okay, like the pleats of a sari. Similarly, okay. Now, the R groups or the acyl groups that are adjacent to the R groups of the adjacent residues will point in opposite directions. Okay, R groups of adjacent residues of amino acids will point in opposite directions. Okay, like this, like a bottle brush, isn't it? How we did the structure of agricane. Agricane is a glycosaminoglycan, a proteoglycan. Agricane is a proteoglycan. We studied about the structure of agricane. It had a bottle brush structure. Similarly, here also the R groups of adjacent residues will point in opposite directions. Okay. The peptide bond, what is the difference between the structure of beta pleated and alpha helix? The beta pleated structure is highly extended. It is highly extended. But the alpha helix structure is highly compact. Okay, understood. The peptide bonds, the residues in the beta pleated structure are stretched. Okay, they are stretched like this, extended. Whereas in alpha helix, they are compact, like a spring. Okay, imagine a spring. When it is compact, it is the alpha helix. When you stretch it maximum, then it will become a beta pleated structure. Got it? It will resemble a beta pleated structure. Okay, one plate of the beta pleated structure. Okay, what is similar in them? That the stability of the structure is provided by the hydrogen bonds, isn't it? Because what I told you in the beginning was that secondary structure is stabilized by hydrogen bonds. Primary structure is stabilized by peptide bonds. Okay. Now, what is the difference between the type of hydrogen bonds in alpha helix and the type of hydrogen bond in beta pleated? What was the type of bond in alpha helix? You remember? Between oxygen of the peptide bond carbonyl and hydrogen of the peptide bond nitrogen of the 4th residue. Okay, this is the type of hydrogen bond in alpha helix. Oxygen of the peptide bond carbonyl, hydrogen of the peptide bond nitrogen of the 4th residue. Okay, this is the type of hydrogen bond here. Why? Because in one turn you have 3.6 residues, isn't it? So this carbon will provide the oxygen, and 1, 2, 3, 4th residue will come here, just below this. So this is the nitrogen, it will provide the hydrogen. and they will form a collinear hydrogen bond. Understood? Why the fourth nitrogen is providing the hydrogen? Because it is the collinear one. Got it? Now again, this is one carbon. It is providing the oxygen. Okay. Now this nitrogen is going to provide. This is what one. This is the. This will be the fourth nitrogen. 1, 2, 3, fourth, and it is going to provide the hydrogen here, and it will form hydrogen bond. Understood? Understood why the fourth residue I am telling? Because these will be collinear. This, if forms a bond with this, it will not be collinear. This will not be collinear. No, this will also not be collinear. Only the fourth residue will be collinear because there are 3.6 residues per turn. Okay, so the fourth residue will be collinear. Understood or not? 1, 2, 3, 3.6 residues per turn. So the fourth residue will be collinear with this. Understood this? This is very, very important. Okay, so this is the type of alpha helix. This is the type of hydrogen bond in alpha helix. Whereas the type of hydrogen bond in beta pleated structure is hydrogen bond between adjacent segments of beta sheet. How? This is one beta sheet. This is another beta sheet. This is the third beta sheet. Okay. Now here it is running in this direction. The peptide bonds are running in this direction. Here this peptide chain is running in this direction. Okay. So what are these? They are anti-parallel. Okay. One is running from left to right. The other is running from right to left. Okay. So they are anti-parallel. All right. Now in anti-parallel sheets, this is the oxygen molecule, this is the carbon molecule. Okay, so nitrogen will provide the hydrogen, and this oxygen is going to come from here. Now these will be collinear hydrogen bonds like these between the two plates. Okay, between the two sheets. Okay, these are collinear, therefore anti-parallel structure is preferred. Why? Because I told you, wherever hydrogen bonds will be collinear, wherever hydrogen bonds will be at 180 degree, that structure will be preferred. Why? Because that is most stable. If the hydrogen bond is bent, then that will be not that much stable. Okay. Now you look at this parallel sheets, all are running in the same direction like this, parallel sheets. Now, can you see the hydrogen bonds? They are slant. Okay, and they slant. Okay, they are slant hydrogen bonds. Slanting hydrogen bonds. Okay, and they slant alternately. Okay, one in this direction, one in this direction. One will be in this direction, which I have missed. Okay, and the other will be in this direction. So, alternately they are slanting. Okay, and since they are non-collinear hydrogen bonds, so this structure is not going to be preferred. Therefore, the structure that occurs most in nature is the anti-parallel beta pleated structure and the right-handed alpha helical structure. Understood? Understood all of you? Okay, got it. What are the other structural proteins present in extracellular matrix apart from collagen? Elastin, fibrillin 1. These are the three structural proteins present in extracellular matrix. What are the three structural proteins? Collagen, elastin and fibrillin 1. Out of these three, which one is the most abundant? Collagen. Okay, collagen is the most abundant protein present in animals, present in the extracellular matrix, wherever. Okay. Now it has a very important role because it provides strength and rigidity to the structure wherever it is present. Okay, so since it provides strength and rigidity, its structure should be we should read its structure focused such that we know what are the different characteristics in its structure that provide it strength and rigidity. Okay. Now, first and foremost, what you should mention is that it is a triple helix. What is it? It is a triple helix. So, what is the structure of collagen? Is it a beta pleated structure or an alpha helix? It is an alpha helix because it is a triple helix. That means it is a protein containing 3 alpha helices. 3 alpha helices will combine together to form collagen. Okay. Now we have seen in nature right-handed alpha helices are preferred. Why? Because their collinear hydrogen bonds form. Okay. Now these 3 helices are individually left-handed. They are individually left-handed, and the suprastructure, the triple helix, the suprastructure that is formed by interaction of these 3 helices, this suprastructure is a right-handed structure. Okay. Therefore, can you see that this structure is right-handed like this? And the individual helices, if we see, the individual helices are left-handed. So, can you tell me what is one important characteristic in the strength of collagen that is derived from this opposite-handedness? Opposite-handedness. How will it contribute to strength of collagen? This opposite-handedness, what is it going to do is, when you try to unwind collagen, when you try to unwind this fibril collagen molecule, the 3 individual helices are going to wind up even tighter. Okay, so it is not possible to degrade collagen like this. Okay, now suppose, see, your hand has a lot of connective tissue. If you ring your hand, ring means if you twist it like this, normally if your collagen did not have opposite handedness, what would happen? It will lose its elasticity. It will turn like that. Okay, but why is your skin so intact, so integral, and so what happened? Why is it not losing its integrity? Why is it always coming back? Because of the opposite handedness. If you ring your hand like this, the skin is coming back, bouncing back, isn't it? You're ringing your hand, twisting your hand, and the skin is bouncing back. Why? Because the collagen molecules present in the connective tissue of your hand, they are prevented from being twisted. Okay, got it? Understood? Why? Because of opposite handedness. Understood, all of you, or not? Do let me know if you guys have understood this. Okay. So you understood why the opposite handedness is important? Okay. Got it. Now this structure of collagen is called a fibrillar structure. What is this? This is the collagen fibril. This is a fibrillar structure. Collagen fibril. Okay. Now per turn, how many residues were there? In a normal alpha helix, per turn 3.6 residues are there. In one pitch, 3.6 residues are there. In collagen, 3.3 residues are there per turn. Okay, 3.3 residues per turn. Okay, and the pitch is almost double. Pitch is almost double that of a normal helix. Normal helix pitch was 0.54 nanometers. Here it is around 1. Okay, got it? That is, it covers around 1 nanometer per turn. Okay, and there are 3.3 residues per turn of this helix. Okay, understood? Now, what are the residues present? The collagen, as we saw, the alpha helix is very compact, isn't it? The van der Waals interactions, the hydrogen bonds, they make the alpha helix very, very compact. Okay. Now, similarly, the alpha helix of collagen is also very compact. It is so compact that in every third residue has to be very small. And that very small residue is which one? Does it ring a bell? The smallest amino acid? Glycine, isn't it? Glycine. What is glycine? It is non-essential, isn't it? Glycine is non-essential amino acid. Okay. Now this glycine is present as every third residue in collagen molecule. So what is the primary structure of collagen? It is represented as GlyXY, GlyXY, GlyXY. Okay, or you can represent it as GlyXYN. N molecules. Okay. Out of this gly is what? Glycine. So every third residue is glycine because the structure is so compact that only a small amino acid like glycine can fit there. Okay. X and Y, what are these? Mostly they are proline and hydroxyproline. Yesterday we have discussed how proline and hydroxyproline are formed, isn't it? How is hydroxyproline formed? Can any of you tell me? How is hydroxyproline formed? What is the enzyme involved? What is the enzyme involved in the synthesis of hydroxyproline and what is the process called? Prolyl residues. Guys, it is very important for those of you who are going to appear for your professional examinations. I expect you to answer this. Prolyl residues will convert to hydroxyproline in post-translational modification, isn't it? This is a post-translational modification. We have discussed what post-translational modification and co-translational processing are, isn't it? You can go back to yesterday's class to understand. What is the enzyme involved here? Tell me fast. And what are the cofactors? Extremely important. The enzyme involved is prolyl hydroxylase. Very obviously. Cofactor involved is alpha ketoglutarate and vitamin C. Very important. Therefore, in scurvy, that is the deficiency of vitamin C, hydroxyproline residues are not formed and therefore the stability, the strength of collagen is deterred. Therefore, the connective tissues are defective and there is delayed wound healing. There is easy bruisability, bleeding gums. All these things occur in scurvy because of defective collagen, because of defective hydroxylation of prolyl residues. And what are the other type of residues present in collagen? Lysyl residues, lysyl and hydroxylysine, lysine and hydroxylysine. Lysine is a iso-like looking at, like lysine is an essential amino acid. Okay, it is an essential amino acid. Lysyl से बन जाएगा hydroxylation में hydroxylysine. Similar enzymes. What are the enzymes involved? Hydroxylation है, so it will be lysyl hydroxylase. Same cofactors. Alpha ketoglutarate and vitamin C. Okay, alpha ketoglutarate and vitamin C, same cofactors involved. Okay, and this is also a post-translational modification. After lysine has been formed, it can be hydroxylated. Okay, now hydroxylysine residues in collagen can also undergo one process that is called, what is this process called? Any of you? Glycosylation. All these provide stability to the structure of collagen. It provides stability to the structure of collagen. Glycosylation means carbohydrates are added. Carbohydrate moiety is added. Okay, got it? Now, one other enzyme called lysyl oxidase is also involved. What is the function of this enzyme? It causes Deamination. What does it do? Oxidative deamination. And what is the cofactor involved? It is copper. So these are the lysyl residues. They will be changed into what? They will be converted into aldehydes. Okay, they will be converted into aldehydes. These are modified lysyl residues. Okay, residues in two different chains, they will interact together and form bonds. And these bonds provide a lot of strength to the collagen molecule and prevent collagen turn. Okay, one more important structure is, this was the fibrillar structure of collagen. Okay, understood? Do let me know if all of you have understood about this structure. Okay, all of you have understood about the fibrillar structure of collagen. Now there is another type of structure that is called the meshwork structure of collagen. How many of you know about the meshwork structure of collagen? Meshwork structure of collagen. This meshwork structure or the network-like structure resembles the mosquito net. What does it resemble? It resembles the mosquito net. Okay, and it is present in type 4 collagen. Type 4 collagen has this structure. Okay. Why? How does it occur? When a few XY regions are obliterated, they are missing. Then what happens? They are replaced by globular regions or holes, isn't it? There will be XY here. Now again here XY is missing, so hole will occur. Again XY will be present. Again here XY will be missing. So what happens? It resembles a mosquito net, isn't it? Where the threads are where XY is present and the holes are where they are absent. Following this. Okay, all of you, this is the meshwork-like structure of type 4 collagen. It occurs in where? Where does it occur? Basement membrane. Membrane. So membrane needs to be something like this. It needs to have pores. Okay, so these pores are provided where, how? By missing XY. Okay. Now one very important term in the structure of collagen is the quarter staggered arrangement. What is the quarter staggered arrangement in collagen molecule? Okay, any of you, do let me know. I need the answer in the comment section. Those of you who are attending, tell me. Most of you are going to take your professional examinations in a few days, so you guys must be knowing. We have done this a lot of times also, quarter staggered arrangement in our super fast sessions, in our revision sessions. What is the quarter staggered arrangement, guys? Tell me. Quarter means 1/4. Staggered means which is shifted. Arrangement is arrangement. So the collagen fibrils, the rod-like structures in the collagen fibrils, they are not placed longitudinally. Rather, see, if they would have been placed longitudinally like this, just one after the other, then there is no staggering. But how are they placed? They are placed staggered or shifted longitudinally, that is about their axis, longitudinally by 1/4 of their length. 1/4 of their length is shifted longitudinally from the other, like this. So this type of arrangement is called the quarter staggered arrangement, and this arrangement provides the What does it provide? The banded appearance of collagen macroscopically. Collagen appears banded. Understood or not? Do let me know. This is the whole structure of collagen that you guys should know. Extremely, extremely important. The structure of collagen is extremely important, guys. Now we are running short of time, so we'll do the structure of hemoglobin in the next class. Okay. And then we'll start with the metabolism of amino acids also, transamination reactions as well. Okay. Now in that we'll start doing from tomorrow. Okay. In the next class we are going to start with glycolysis. Okay, that is glucose metabolism. We are going to start with glycolysis, and then we'll continue gluconeogenesis, HMP shunt, Krebs cycle, and all. Okay, glycolysis is more or less an easy topic, so we'll do it fast. Okay, understood or not, guys? Do let me know if you have understood. If there's any other doubt, then I can take it. Okay, and I have noted down all of your suggestions. I'm going to base my classes depending upon that, because whatever you guys demand, howsoever whatever you guys want to study, that is going to be our pattern. Okay, because these classes have to benefit the maximum students. Okay, all right. Understood the structure of collagen, the quarter staggered arrangement, banded appearance, the alpha helix, the beta pleated structure. Okay, difference between primary, secondary, tertiary, quaternary, the different types of bonds that stabilize different structures. Secondary structure, extremely important to know about alpha helix and beta pleated structure. Very, very important. Okay, difference between conformation and configuration, extremely important. Okay, guys, so with this, I think we'll wind up this session on proteins, and we'll meet in the next session after a few minutes on glucose metabolism, where we'll be starting with glycolysis. Okay, all right, then you can take a short break, and then we'll meet again in the next session on glucose on this channel itself, Unacademy Future Doctors. Do subscribe to this channel if you haven't yet, and do share it amongst your friends so that we get a lot of more interactive, intelligent, smart students here with whom we can share knowledge. Okay, all right, thank you, bye bye, see you soon.